cell, and we have gone up to, I hope we have completed, uh, yes, this we are talking regarding cytoskeleton, then mitochondria, and then this endoplasmic reticulum, and this uh, yes. Now coming to this Golgi bodies, my screen is visible to everyone.
Yes,
Yes,
sir.
Yes,
sir.
sir.
Yes,
Okay,
sir.
Yes,
Yes,
sir.
sir.
okay, thank you. So
No, sir.
okay,
No, sir.
uh, my screen is not visible.
No, sir.
No, sir.
No, sir.
No,
No,
sir.
sir.
The screen
Screen
is
is
not
visible.
visible, sir.
No, sir.
No, sir.
Oh,
No,
Sir,
okay,
sir. It is not visible.
you are visible, sir.
okay. Let me see what happened. Just do it. Okay. I think it should be visible. Share. Mhm. Just click it. Now visible?
Yes, sir.
Yes, sir.
Yes,
Visible.
sir. It is available.
It is visible.
Okay. So, okay. So, okay. So is it visible, right?
Yes,
It
sir.
is. It is
Yes,
visible.
sir.
So, okay. So please mute your microphone. Please mute your microphone. Okay. So apart from that, mitochondria and endoplasmic reticulum, in the cell you will find some pancake-like things are there that we call them Golgi bodies, or this, and this has two phases. One is your trans phase, and another one is your cis phase. So see, there are two phases of this Golgi bodies, and these are flattened sacs, and Camillo Golgi is responsible for this discovery of this Golgi bodies. Okay, and mostly the transport vesicle, they get attached to this your Golgi bodies, and by this the transportation work is carried out. Okay, with the help of this transport vesicles. This is a transport vesicle. Okay, uh, where is my, yes, transport vesicle, and see how the transportation is happening. So it's like pancakes. So they can modify, they can sort and package these molecules that are coming from this endoplasmic reticulum. Okay, so different proteins, different other molecules, they can be. Now you see how these molecules are coming from. So this is your nucleus, and this is your endoplasmic reticulum. These are raw endoplasmic reticulum, and now this is coming to these Golgi bodies, and now it is coming out of the cell. Okay, this is the process by which substances, those are um, present in the cytoplasm, or they may be present inside the nucleus, they can move through this endoplasmic reticulum and move out of the cell. Okay, so this is how the Golgi bodies, they help in the transportation, okay, through the vesicles. And now coming to another organelle, that is your lysosome, and you know this is called the suicidal bag of the cell. Yes, yes, suicidal bag of the cell. And this in fact, why we call this a suicidal bag, can anybody tell?
Because it digests the
Its own
They
cells.
release hydrolytic enzymes
Okay,
in order to digest the
okay. So it
The foreign
contains
pathogen digest the
yes,
antigen,
yes.
foreign pathogen, whatever, which is
Okay,
not
so
necessary to our body.
okay, then any other things?
Yeah, actually, the worn out cells, they are actually digested by this lysosomes. They
Yes, yes, very
encircle it
good.
and
Very good, very good. In fact, see, just like certain things are when not used by the cell, they can be degraded and again they can be recycled. Okay, for other purposes. So in that case also lysosome plays a vital role. For that the lysosome is there, and you know Dubé is responsible for this discovery of this lysosome. Okay, so they break and open, so release the enzymes and they recycle the cell parts. Okay, now see, they can take food by phagocytosis and they can get rid of wastes. Okay, and you know all these things, and this cilia and flagella, those are present in a lower group of organisms. You will find the arrangement like 9+2 arrangement, this arrangement. Okay, and what is the main role of this cilia and flagella? Can anybody tell?
Movement.
Mobility for the locomotion.
Movement
Very
required.
good.
For the locomotion.
Yes, 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 yes. Now this is the cilia. These are very short and numerous. These are the cilia, and these are your flagella. Okay, these are longer and they are less. These flagella are less number in cell, whereas cilia are numerous in cells. Okay, so this is how this is the movement of a spore, and this is like a paramecium. Okay, you know these are the cilia, so they can help in locomotion or these things. Okay, and these are the cilia, those are present in our respiratory system. Okay, and now coming to vacuole, you know the vacuole is mostly present in case of your plant system, and there is a covering over the or the outer layer of this vacuole. We call them like tonoplast, right? And oh my god, they are very small. Anyway, so in plants they store cell chaff, and they include the storage of sugar, protein, minerals, lipids, and waste as well salt, water, and enzymes also. They are present inside these vacuoles, and this is a contractile vacuole. Who can tell what is the role of a contractile vacuole?
It helps in osmoregulation.
Very good, very good. Osmoregulation. So this is this your macronucleus. This is your micronucleus. These are food vacuoles. Okay, and this is the contractile vacuole. You see, most of the time these organisms, when they live inside the water, okay, due to the process of this maybe diffusion or so the water is coming inside the body of the organism, and once this maximum water is there inside the body, it need to be released. Okay, so through this contractile vacuole they can release the extra amount of water that is there inside the body of the organism, otherwise it will prostrate. Okay, so in paramecium, so it regulates the water intake and pumping out and make and helps in homeostasis. Okay, and helps in keeps the cell out of bursting. Okay, now coming to the next organ, that is your chloroplast. Okay, and the chloroplast, those are mostly found in plants, and in some lower organism also you can find like bacteria chloroplast or the things, but mostly these are found in your higher plants. Okay, and you know the function of this chloroplast. Mostly they,
Photosensitive.
yeah, they produce this carbohydrate type of food material from carbon dioxide, water in presence of sunlight in chloroplast, right? So there are two kinds of reaction. What are the two kinds of reaction that is happening in chloroplast?
Light
One,
reaction, dark reaction.
light reaction, dark reaction.
okay, so this dark reaction and light reaction that is happening in chloroplast. In which part of the chloroplast the dark reaction happens?
Stroma, dark reaction stroma, light reaction.
Okay, Why the dark reaction is taking place in stroma and this light reaction in grana? Can
Light
anybody tell?
reaction.
Why light reaction is taking at grana and the dark reaction in stroma?
Because
So
grana
grana
contain
contains
chloroplast.
the
Grana.
chlorophyll pigment, I guess. So
Okay,
they can trap
pigments.
the sunlight, whereas in stroma it's
Very
not.
good, very good. These light harvesting compounds or these light harvesting molecules, these LHC molecules or proteins, those are present in grana, so they are responsible for this light reaction. Okay, but however, the enzymes, those are responsible for carrying out this Calvin cycle, that is they are present in your stroma. Okay, so due to the presence of these enzymes, this dark reaction is taking place in stroma, not in grana. Okay, so Schimper has observed these organelles in cell in 1883. However, this Konstantin Markovsky has told regarding this chloroplast, but the discovery was done by US Schimper. So this is a double membrane surrounded organelle. Okay, so there is outer membrane and there is inner membrane. These are your thylakoids and these are the stroma. This is the stroma, and this outer membrane is smooth, and this inner membrane is modified to like sacs-like structures. We call them like this thylakoids, and thylakoid in sacs are called grana. They are interconnected. Okay, and this stroma is a gel like material, and now it contains its own DNA. So these are semi-autonomous like organelles, and they contain the enzymes and pigments for photosynthesis, and we never find these organelles in case of your animals. Okay, now though we have discussed ribosome yesterday, but you can see some more slides. Ribosome is the smallest organelle. Which one is the largest organelle? Which one is the largest organelle?
Nucleus.
Very good. And this is yes, but this is ribosome is one kind of organelle which is not surrounded by membrane, and ribosome is made up of what? You can tell.
RNA and proteins.
Yes.
Ribonucleic.
Okay. So there are these are the larger subunit. This is the smaller subunit, and it is made up of your proteins mostly, and these are attached to your endoplasmic reticulum, and sometimes these are also free floating in the cytosol. Okay. Now I have told you that it has different compartments like this. In the larger subunit, you have your acceptor site and this exit site and this polypeptidal sites. Okay, so the amino acid first come to this site and then come to here where the polypeptide or peptide is produced, and then it moves through the exit site. Okay, so there are there are two kinds of these ribosomes, that is 70S and 80S. Okay, and we'll discuss that in the later stage. And the another organelle, that is your peroxisome. These are vesicles. This contains enzymes, and mostly they use oxygen and produce hydrogen peroxide. Okay, and these are very much abundant in your liver, and they produce bile salt and cholesterol and break down these fats. So this is all about your cell organelles that is present in a cell. In very short span of time, I discuss all these organelles, and today we will switch over to the most important thing that is present around a cell, that is your plasma membrane, and what is its role, and how it is helping in different functions. We will see that. Okay, now coming to the can anybody tell what is the role of this plasma membrane? What will happen if there will be no plasma membrane will be there, right?
Transportation is not
For
occurring.
transportation.
For transportation.
Transportation.
Okay, one is transportation. Then other thing.
It is a selectively
Sir,
formed.
Selectively permeable membrane.
selective permeability.
Okay. Then
Acts as physical barrier.
Maintains the shape.
okay. Then so there are certain proteins
Cell signaling from
yes, yes. There are certain proteins attached to this plasma membrane. They help in cell recognition. They can identify which one is a foreign body or something like that. Okay, there are carrier proteins. There are channel proteins. Okay, so those things are there in the cell, in the plasma membrane or over the plasma membrane. So those help in mostly transportation and recognition, and also in cell communication, right? So how the cell membrane or the plasma membrane is looking like, and there are certain models, and regarding these models, this is a Daniel Dabson model. Okay, this is called your sandwich model. Then this Robertson's unit membrane concept. Okay, then this Singer and Nicholson proposed this fluid mosaic model. Which model is the fluid mosaic model? Which model is the most accepted model?
Fluid
Fluid mosaic.
Why? Why fluid mosaic model is mostly accepted?
Because it explains the permeability, how the cell can do it.
And also you'll find, let us see why we accept this Singer Nicholson proposed fluid mosaic model as the best model to explain the plasma membrane. Okay, so this Gotter and Grandel, they show this plasma membrane, and they told that this is made up of phospholipid. This is your polar hydrophilic head, and this hydrophobic tail. Okay, and so this is your phospholipid bilayer. So this is your so it doesn't explain why these non-lipids are permeable. And then coming to this Daniel Dabson's sandwich model, you'll see. So this is your sandwich model. Okay, so this is your protein, and this is your phospholipid. Okay, and then coming to this Robertson's unit membrane concept. Okay, so this is your Robertson's model, and here in this Singer Nicholson model, you'll find there are certain proteins which are embedded inside this phospholipid layer, and you see these are your yes. So these are your proteins. These are your proteins. Okay, and these proteins, some proteins are found inside this membrane, and while some are coming out of that. And these extrinsic and intrinsic proteins are there. And you see, whenever we take this electron micrograph with a certain molecular knife-like structure, and we have seen these proteins are here, you see. Okay, so as per this electron micrograph structure also, it supports this this fluid mosaic model. Okay, so for that reason, see, so special knife is used to cut this membrane into half, and this, after applying this carbon platinum coating, you can find these structures like this. The protein are here. Okay, so and see, this is your fluid mosaic model, and this is your protein. See, and in this model you will find that these proteins are associated with certain carbohydrates, like these oligosaccharides are there, and you see, and these cholesterol molecules are also there. These are your glycolipids, lipid attached to this carbohydrate. This is called glycolipids. Similarly, you can have glycoproteins. right? So the plasma membrane is like that of the olive oil in body temperature, and due to the presence of your unsaturated fats, and this cholesterol affects the fluidity, and these membrane proteins, okay, different proteins are there present over this plasma membrane and inside the plasma membrane. So some are attached with this lipid, and some are attached with this carbohydrates, okay. So those are there. Now this is the structure of your plasma membrane, where this is like carbohydrates, and this is your the cytoskeleton filaments, okay, and this is glycolipid lipid, like carbohydrate attached to this lipid, and this is glycoprotein, okay, protein carbohydrate attached to this protein, we call that as glycoprotein, okay. So this is your cholesterol. So these things, they constitute your plasma membrane. So, and yes, this phospholipid bilayer is there, hydrophobic head, hydrophobic tail is there, cholesterol there, this transmembrane intrinsic integral protein are there, then extrinsic or peripheral proteins are there, cytoskeletal filaments are there, then carbohydrate chain, glycoprotein, glycolipids, all these things are there in a plasma membrane, okay. Now yes, so in the plasma membrane there are certain protein associated with that. And what are the different kinds of protein? Basically there are 6 types of protein present with transportation, okay. Now they can be receptor proteins, okay, they can recognize the signals here. And similarly there are proteins which act like enzymes, okay, and they're responsible for cell recognition, like this cell recognition. Then there are proteins which we call them attachment protein. Then there are intercellular junction proteins, okay. So we'll see one by one. So coming to this, so these are your intercellular junction proteins are there. See, you know, this tight junction, gap junction, this plasmodesmata, you know, desmosomes, you know, all these things. So we'll see how these proteins are helping in carrying out different functions, okay. Now coming to this, in the transportation of molecules, right? So these are 2 types of protein. One is called a channel protein, another one is called a carrier protein, okay. So you see the channel protein here, the channel looks like uniform, and it is open, right? It is open at both the ends. This is the channel protein. However, the another protein is there, we call them like a carrier protein, okay. So this carrier protein, you see, it has different shape, and you see this side is open, whereas this side is closed, okay. So whenever there is a change in the conformation of this protein, this side will open and this side will be closed. So we call them as like carrier proteins. So they change their shape depending on the situations, okay. We'll see them. And these receptor proteins, they bind to the signals, okay. So these are the signals. They may be some chemokine or cytokine, or maybe some any other substance, okay. And they can help in recognizing certain situations, okay. Then there are enzymatic proteins. So these proteins, they help in carrying out different enzymatic functions, okay. So in enzymes, what is the site in enzyme that is responsible for carrying out different reactions?
Active sites.
Very good. There are
Active site.
active sites, and that, of course, when you will be reading this enzyme biomolecules, you will be knowing how are the active site, what is allosteric site, okay, the inhibition, all these things you will be learning. So these are enzymes are nothing but proteins, right? You know, these apoenzyme, holoenzyme, prosthetic group, all these things, you know, and also you will get more knowledge regarding that in your biomolecular enzymology class. Then there are certain proteins, we call them like cell recognition proteins. Okay, so these are glycoproteins and glycolipids. These are extracellular surface, and they work.
ये सब बात आप बोलने के पहले तो मैं अपना प्रपोजल रखा था कि सेल कैसे बढ़ाएंगे.
Yes.
हाँ, वो बोलने के पहले मैं अपना ये रख दिया, वो इसलिए रखा हूँ.
Okay, okay. So yes, so these are your glycoproteins and your glyco. Okay. So now there are certain proteins which are your attachment proteins. Okay, so these are your attachment proteins and they attach to a cytoskeleton. Okay. Now, and the last, so there are different proteins like intercellular junction proteins, and you know there are tight junction and gap junction, and see, and now
बिल्कुल नेशनल नहीं, इंटरनेशनल लेवल पे नहीं.
after going into this plasma membrane transport, these molecules, they move across the plasma membrane.
अंकुर वाला भी मेरे पास आया था.
What you are disturbing? Why you are disturbing?
Sir, your voice is not at all clear.
Voice is not clear?
No, sir.
I can hear you, but how can't you hear me? Anyway, so just is it audible?
Yes, sir. Yes, sir.
Okay. Okay. So the plasma membrane responsible for transport. Okay, and this molecules, they move out of the cell or into the cell through this plasma membrane. Okay. So there are two kind of transport system, you know. One is your passive transport, another one is your active transport. Okay. So what is passive transport?
Sir, it do not need
Does not
ATP, sir,
require energy.
It do not require energy.
So yes,
Along the it passes along the concentration gradient.
very good, very good. And what about this active transport?
Against the concentration
Requires
gradient.
energy and it's an uphill transport.
Energy is required.
Okay, so when there is a passive transport, what is the need of this active transport? So when there is a passive transport mechanism is there, why we need active transport? Because in active transport, lot of energy is utilized.
But there are some molecules which are not soluble through the cell membrane. So
No, anything that is going with the through the plasma membrane, that must be soluble. Nothing solid can pass through this plasma membrane. Okay, you see, there are certain situations, just like imagine a plant. Okay, so the roots, they are taking these minerals through this root. Okay, and the concentration of these minerals inside the plant cell is much more higher than that of the surroundings, but still then it needs more. Okay, so how they can be transported into the from the root to the apex of the plant? Okay, so in that case you need some amount of energy that is utilized, and that kind of transport is taking through this active transport. Okay, similarly in case of these organisms, those are living inside the water bodies. Okay, so in that case the outside environment that contains a lot of water, whereas the concentration of the solute is more inside this organism. So in that case, due to this process of like osmosis or something, it will take up water, and this salt will come out of that, but that is not happening, and it is happening due to your active transport. Now coming to the different forms of your passive transport, here you don't need any kind of energy, or ATP is not required, and there is diffusion, there is facilitated diffusion, and there is osmosis. So these are basically the three processes through which your passive transport that takes place. Okay, now coming to this diffusion, you see, through this molecule can directly go through this plasma membrane, and from higher concentration to lower concentration, these molecules move. Okay, so and here the net movement of this molecule that happens, and diffusion rate is related to temperature, pressure, and this state of matter, size, concentration gradient, and surface area. Yesterday I was telling you that cell has a smaller size and a larger surface area, relative surface area, and it is the process of diffusion that depends on this surface area. Okay, now this is how from higher concentration to lower concentration these molecules are moving. Now through this process of this diffusion, the gases, oxygen, carbon dioxide, the water molecules, the rate is very slow, and these lipids and lipid soluble molecules, hydrocarbons, alcohol, some of the vitamins, and some non-charged molecules, they can pass through the membrane. Okay, now I'm audible, right?
Yes, sir.
Okay.
Yes, sir.
Okay.
Yes.
Okay.
Yes, sir. Yes, sir.
Okay. Now, why this diffusion is important to the cell and humans? It is responsible.
Sir, are you showing the slides?
Yes. Yes.
But it's not visible to me.
It is not visible.
No, sir, you are visible, not the slide.
Is it not visible to you or to everyone?
Don't know.
No, no,
about
it's visible,
that,
sir. It is visible.
It is visible, sir.
Sir,
It is visible, sir.
Clearly visible.
Okay. Thank you. So this diffusion process is taking place during the cell respiration, in the alveoli of lungs, in capillary, in red blood cells, and for this when you are taking some kind of medication also, this diffusion process is much more important for us. Okay, now coming to this facilitated diffusion, when there is helping in this process, so we call them like facilitated diffusion. You see here, this is your normal diffusion, okay, and here this is facilitated diffusion. When the size of the molecule is larger and when the rate of this diffusion is faster, that can happen through this facilitated diffusion process. Okay, so here this the same thing happens. It happens on the basis of this concentration gradient. So molecules, they move from higher concentration to lower concentration with aid of this channel protein or carrier protein. Okay, so this is your carrier protein or channel protein. We'll see. So through this facilitated diffusion, the ions like sodium, potassium, and these chloride ions, these sugar molecules, these amino acids, and these small water molecules, they can small molecules, water soluble molecules, and water in a faster rate, they can move through this plasma membrane. Okay, now this is your channel protein, okay, and this is your carrier protein. Okay, and now this is happening through this process is called your facilitated diffusion, because the diffusion is happening through the help of another protein. Okay, yes. So these channel and carrier proteins are specific, and these channel proteins allows this ion, small solute, and water to pass through this plasma membrane, and these are also responsible for carrying out taking this glucose and amino acid inside that. Okay, and this facilitated diffusion, the rate of this diffusion is limited by the number of this channel or carrier proteins present over the membrane. Okay, now there are specific type of facilitated diffusion. See, sometimes what happens, like two molecules, they can different kinds of molecule, they move in different direction at the same time. You see here is one amino acid that is red in color, and this sodium ion that is your yellow in color. And you see when they move, so they move in opposite direction. Okay, so when the transport of two substances that takes place in the same time but in opposite direction, we call that as counter transport, and the protein are called antipodes. Okay, here also you don't need the help of ATP, whereas there is another process that is called co-transport. Okay, so in co-transport, what happens? You see, the same time, okay, these transport of two substances taking place in the same time in same direction. Okay, so in same time in same direction, and that process is called co-transport. Okay, and the protein that is carrying out this function, we call them like sympod. So is it clear what is counter transport and what is co-transport? Okay, so in co-transport, the movement of these substances takes place in same direction in same time simultaneously. But in counter transport, though the transport happens simultaneously, but they are in different direction, in opposite direction. Okay, and the carrier protein in earlier case it was sympod, and in case of counter transport it is called antipode. Okay, and sometimes there are certain channels which are there is a gate like things, we call them like gated channel proteins, and these gated channel proteins here now it is closed, and due to some kind of signals and this channel is open, now there is movement. So these are your gated channel proteins. So you can have your antipodes, you can have your sympod, and you can have your gated channel proteins. Okay,
Sir,
yes.
sir, antipode is a co-transport.
Now see, antipode is counter transport.
It is also called as co-transport.
See, yes, in case of this, see, in case of co-transport, both these molecules, they move together.
Yes.
Okay.
Yes, sir.
Now, why facilitated diffusion is important? Okay. So cell obtain food through the respiration, neurons communicate, and the small intestine cell transport food to blood streams, and muscle cell contraction, and these are the functions that is happening through this yes, facilitated diffusion. Now coming to the last part of that passive transport, that is your osmosis. Okay, here the movement of this solvent takes place. Basically this liquid water is the universal substance that moves directly through this membrane, that is called your osmosis. Now see, so here the diffusion of water takes place through the semi-permeable membrane. Okay, now see, so if there is a semi-permeable membrane here and this side is more concentrated than this, then after some time you will find that this solvent or this water, it will move to this side. Okay, why? In order to, yes, can anybody tell why the water moved to this side?
Maintain the equilibrium.
Very good, very good. So the water will move from this side to this side in order to maintain the equilibrium. Okay, and once the concentration is equal in both the sides, then there will be no movement of water to this side. Okay, so this is responsible to remove the water. Those are produced through respiration, and these kidney cells form this urine. And now coming to this tonicity, there are three situations depending on this tonicity, and it depends on the solute concentration of the solution of the outside cell. So there can be three kinds of solution. One is isotonic, then another one is hypotonic, another one is hypertonic. Okay, so in an isotonic solution, you'll find that the concentration of the cell and the outside the cell, that is same. Okay, so here this is 10% solution and this is also 10%. So there is no change in the shape of the cell because it is in isotonic solution. Okay, so there is no transport of water molecule into or outside of the cell. Okay, now next is so there is no net movement, so there is no net charge and no net change in the shape of this cell. Now coming to a hypotonic solution, when the concentration inside this cell is see, it is 20% and it is 10%. So the concentration inside the cell is more than that of the outside of the cell. So what will happen? Then this solvent or this water, they'll move inside the cell. Okay. So the cell will swell, it will gain water, and if the concentration is much more here, then the cell will burst. That is called your hemolysis. Okay.
Endosmosis occur.
Yes. So in case of plants, the rigidity of the cell wall and this turgor pressure is due to this. Now coming to this hypotonic solution, when the concentration is here much more higher than this, then what will happen? The water from the cell, it will move out of the cell. So there will be a shrinkage in the cell size. Okay. So there will be a shrinkage, and this thing we call like plasmolysis, right? So here, what is this? There is no change. So this is isotonic, and this is what happens. So this is hypotonic, where the water is coming inside the cell and the cell size is increasing, and ultimately it is there is burst of cell, and here the cell is shrinking. Okay, the concentration is outside is more than that of this cell inside, so the water is coming out of the cell. Okay, so there are three kinds of passive transport, that is diffusion, facilitated diffusion, and osmosis. Okay, so and we told you regarding this hypotonic, isotonic, and hypotonic solution, and how the cell size and the cell shape is changing due to a change in the tonicity or concentration of this solvent inside the cell. Okay, now coming to the next, of course today we will not able to finish this portion. We will be continuing with this, that is your active transport. Okay, so in active transport, as you know, we need lot of energy for this transport. Why can anybody tell?
Yes, sir.
Because
It's
it
against the
Because it's
concentration
against
gradient.
the concentration
Okay,
gradient.
it is happening against the concentration gradient, right? So you need some energy, and the energy basically in case of
Because materials are moved against the concentration.
Very good, very good, very good. So there are there is exocytosis, there is endocytosis, of course there is receptor mediated endocytosis. We will learn them one by one. And phagocytosis. What is phagocytosis? So here ATP is required. Okay, now these molecules, they move, so this is your diffusion. Okay, there is no need of any specialized thing. And this is your facilitated diffusion, you need carrier protein or channel protein. And you say this is your active transport. Here the concentration of this substance is less, here it is more, still then this substance is coming inside the cell. Okay, and this is due to active transport. And in the next class we will deal with the active transport. Okay, now students, those who think that they have not responded to the attendance, they can send the message through this chat box. And thank you for joining today. And if anyone is unable to hear my voice, or there is a problem in audio, he can contact me. I will give you the slides so that he can go through the slides, and if he or she will have any kind of problem, then he can contact me. Okay, thank you.
Thank you, sir.
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 Thank you,
Thank
sir.
you, sir.
Welcome. Welcome.
Thank you, sir.
Thank you so much, sir.
Thank you.
Welcome. Welcome.
Thank you so much,
Go to
sir.
the next class. Yeah.
Hello.
Hey,
Hi. Is there is any link of next class you have?
I don't know, but
No,
I think
I don't know.
the same class is there again. I mean,
Yes.
Elisa
Huh.
just
Yes,
only.
yes, yes.
But why the sir is end the class here? I don't
Please
know.
contact.
Okay, okay.
Why is everyone leaving?
I don't know, sir. I called sir, and sir told that give me 5 minutes, I will join. But everyone leaving and chatting on the group.
Okay.
Hello,
Yeah. Okay. Actually, I was wondering that. Okay,
sir.
classes started.
Sir is here.
Yeah.
So am I audible to everyone?
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir. Yes, sir.
Yes, sir.
Yes,
Okay,
sir.
okay, thank you. So whenever there will be any kind of problem, audiovisual problem, you see, this is happening due to sometimes due to this network failure, or sometimes due to slow internet. So you have to be aware with that, because always the speed of the internet doesn't remain the same. Okay, you know very well these things. That is the part of this online system. So sometimes the voice is not audible. So please be aware with that. And if you have any kind of problem in that thing, then you can uh, write an email to me uh, regarding that problem, and I will take the necessary steps how to cope with that. Okay, so all the students, those are there in the first class, are now joined, I hope.
Yes,
Okay,
sir.
Okay.
Yes, sir.
Yes, sir.
Yes, sir.
So, okay.
Yes, sir.
So in the earlier class, someone was complaining regarding this audio. Who was that? Who was that? Don't say. If it is a genuine problem, then I'm ready to address that.
Sir, it's me. The slides are not visible that time.
Mm-hmm.
My friend told to log in another time, then it's okay.
Okay. Okay. Now regarding this voice, someone was telling that the voice is not audible or something like that. Okay, no problem. Don't worry. So let us move to this. Look into these slides. No, this is not. Another one is there. So my screen is visible. Hello.
Yes, sir.
No, sir.
No, sir, it's
No,
not.
sir.
No, sir, it is not visible.
It's not visible. Let me see.
No, it ain't.
Okay. Now, of course, it may be. I don't know why you can share that. Just a minute. Now, is it visible? No.
No, sir.
No, sir. No, sir.
सर
No, sir. No, sir.
No, sir.
No, sir.
Is it visible?
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes,
Yes,
sir.
sir.
It is visible now.
Yes, sir.
Okay, that is fine. That is fine. So we have discussed regarding this passive transport. Now let me ask you certain questions. Then we'll move forward. Okay. So why this is called passive transport? Because here there is no use of
No requirement of energy.
okay external. So of course without energy there cannot be any kind of movement. But the energy, where from we are getting that energy?
Mitochondria.
No, see, whenever there is any kind of process that is happening, without application of any external energy, that this thing cannot be possible, right? Everything will be at its inertial position. But when there is a movement, the energy is provided by the concentration gradation, right? So when one side the concentration is much more, then in order to maintain equilibrium, these molecules they pass inside the cell, or the something coming out of the cell. Okay, so the energy that is provided, that is not provided by the cell, right? Okay, so suppose, do you agree to that or not?
Yes, sir.
Yes.
Yes, sir.
So the energy that is required, of course ATP is not required, but the energy that is required, that is provided by the concentration gradation, right? So when the concentration, so the molecule, they move from higher concentration to lower concentration, whether it is osmosis or it is facilitated diffusion or diffusion, right? So when there is a process like diffusion, what is the need of this facilitated diffusion? Who can tell? If water can move through this plasma membrane through the process of diffusion, what is the need of this facilitated diffusion?
The solid
Charged
molecule.
molecules move through facilitated diffusion.
So through this normal diffusion, the small molecules and even water, the rate of this movement is very slow, okay, or very less. So for a rapid movement or for the movement of these larger molecules, you need to have a facilitated diffusion. And this facilitated diffusion is done by what? Which is responsible?
Carrier protein.
Carrier
Okay.
protein, the channel
Carrier protein, or this you call this channel protein, right? So what is the difference between a carrier protein and a channel protein?
A channel protein has a pore, while carrier proteins do not have open pores.
Can you repeat your answer, please? Yes.
Channel proteins have open pores, while
Yes.
carrier proteins do not have open pores.
Very good. Very good. So you see,
carrier proteins bind specific molecules that to be transported to the next side of the membrane.
okay. Thank you. So, see, this is a channel protein, right? And this is a carrier protein. Of course, the function is same, right? Their function is same. So, in absence of this carrier protein or channel protein, how these molecules, they are moving inside this plasma membrane? In absence of these carrier proteins or channel proteins. Here you see, there is no carrier protein or channel protein. Still then, these substances are there coming inside. Maybe, my God. Okay. Yes. So, you know, these things, they can move in a higher rate, and whenever there is the absence of these carrier protein or channel protein, still then through these intermolecular spaces, these small molecules or these little amount of water, they can pass through this. Okay, so that is your normal. You see, your plasma membrane is not like a membrane that cannot admit like, or that cannot allow the passage of all the substance. No, this is a semi-permeable membrane. That means it is permeable to something and it is not permeable to some other things. Okay, so we call them like semi-permeable, right? And you have seen this counter transport and co-transport, this antiport, symport, and your gated channels. And we have completed these things. Now osmosis and this tonicity also we have completed. Now, so tonicity is clear to everybody?
Yes, sir.
Okay.
Yes, sir.
What
Yes,
is
sir.
what is an isotonic solution?
Yes, sir.
What do you mean by isotonic solution?
When the concentration on both
The concentration are equal, sir.
One with equal concentration of solutes and a solvent.
Is it?
Equal
The con-
concentration.
the
Equal
con-
concentrations both inside the cell and outside the cell.
That is fine. That is very good. What is your name?
Sudarshana.
Okay, thank you. So what is one hypotonic solution?
Concentration is more. It means solute is more than
Solute
solvent.
concentration.
Just 1 minute, please. Arun. Uh-huh. Uh-huh. Arun. Arun, what class notes for a So, what is an hypotonic solution?
So the concentration of the solution inside the cell is lesser than the outside the cell.
So that is your hypotonic. So if you keep one cell in hypotonic solution, what will happen?
Endosmosis occurs. Cell inside the cell to out the cell, water will move.
Water will move from cell to the outside, right?
Outside.
So
Yeah,
what
and
will
the cell
happen?
will shrink.
The size of
Hemolysis.
yes,
Hemolysis of cell.
yes. There will be shrinkage in the cell size, right?
Shrinkage. Yes.
Okay, the volume of the cell will decrease. But if you keep one cell in hypotonic solution, what will happen?
Bursting of the cell.
Okay, why? Because the water molecules they will enter inside the cell.
Inside the
Okay.
cell. Yes.
Okay. So we have discussed regarding this tonicity. Now, these are some of the things we gone through this. Now coming to this active transport. See, in active transport, you have heterocytosis, you have endocytosis, and in endocytosis, you can find this phagocytosis, pinocytosis, and receptor-mediated endocytosis. Okay, so you know that during this active transport, you need ATP. Okay, so cell has to invest some energy that will be required for the transport of the substance opposite of its concentration gradient, right? So the movement of the substance will take place from lower concentration to higher concentration. So for that reason, you need certain amount of energy, and that energy is provided by the ATP in a cell. And also it also requires certain carriers. We call them like pumps. So among different pumps, so there are three methods to move solute against this electrochemical gradient. One is your coupled transporters, then ATP driven pumps are there, then light driven pumps are there. Okay, so we'll see how these things are taking place in a cell. See, what is the importance of this active transport? Okay, so through this process of your active transport, these important molecules like ions, amino acids, sugar molecules, these nucleotides, they can move, and the cell can get rid of unwanted molecules. Okay, just like sodium from urine in kidney. Then it also maintain internal condition different from that of the environment. Okay, and it regulates the volume of the cell by controlling the osmotic potential. It also have able to control the cellular pH, and it is also helpful in maintaining or establishing sodium potassium pump. Okay, so here this sodium potassium pump, you will see that they work in that they move in opposite direction. Okay, you'll see this diagram. See here, this is a carrier protein. Okay, so in this carrier protein you will find two kind of conformation. Okay, see here, these potassium molecules, they are moving from this side to this side, and these sodium molecules, they are coming from this side to this side. This is your inside the cell, this is your extracellular fluid. Okay, see here the concentration of this potassium ion is less, and here it is more. Okay, still then it is moving. And similarly, the sodium ions are here less, but it is still then it is moving outside. Okay, so this is happening against the concentration gradation or gradient. So you'll see that now so here what will happen, now this side is open, so what will happen, these ATP molecule will bind here. I don't know why this is not working. The animation should work. Anyway, see, now 3 molecules of now they are coming, and this 2 molecule of potassium they are coming, and this is dephosphorylized, and this side is open. Now this is, and it is again happening for another cycle. Okay. So you see 3 sodium ion move out of the cell, and then 2 potassium ions they move inside the cell. Okay, so when these potassium ion they are moving into the cell, okay, this is dephosphorylized, and once this is these your potassium ions, when they are when the sodium ions they are coming inside, this is dephosphorylized, and this ATP is utilized. So what is the role of this ATP here now? Can anybody tell?
ATP opens the gate to move inside.
Okay, so the amount of energy that is utilized in changing the conformation of this protein, right? So the protein, the gate is open this side, and now it will be open this side, right? So this ATP is used to establish this electrochemical gradient across the neuron cell membranes, and this ATP is helpful for the conformational changes to protein by adding then and taking out this phosphate group. So whenever the phosphate is attached, we call this as phosphorylation, and when this phosphate is taken out, this is called what?
Dephosphorylation.
Can anybody tell which is the enzyme that is responsible for this phosphorylation? Which enzyme? No. ATPase. What is its role? You see, when we say about certain enzymes, how the enzymes are named, these are named on the basis of the type of reaction or on the substrate on which it is working. Okay, so when you are telling ATPase, okay, so when there is a breakdown, you use this ASE, okay, ATPase, nuclease, okay. So what it will do?
Sir, converts ATP to ADP.
Okay, it breaks down ATP to ATP and inorganic phosphate, and during the breakage of that bond, a lot of energy is produced. Is that clear? Okay, so
Yes, sir.
so for this, and when one phosphate group is attached to some other compound, like you have glucose and it becomes converted to glucose 6-phosphate. What is the enzyme?
Kinase, I mean hexokinase.
Very good, very good. So when you are adding one phosphate molecule, then always the enzyme is kinase, and depending on this substrate, you call it hexokinase or glucokinase or fructokinase or something like that, and in general you call that as hexokinase, right? And when you remove one phosphate group, what is the enzyme? What is the enzyme?
Phosphatase.
Phosphorylase.
Very good.
Phosphorylase.
Phosphatase. Okay. So this is your sodium potassium pump, and it is very important on the examination point of view. Normally, this is a short question asked to describe the principle or mechanism of this sodium potassium pump. Okay. So whenever there will be a question like that, what you have to do? You have to draw this diagram. Okay. You have to draw this diagram and you have to write these couple of points. Okay. Can you do that?
Yes, sir.
Yes,
Yes.
sir.
Yes, sir.
Yes, sir.
Okay. Now, similarly, there is another pump that is called your calcium pump. Do you know where the calcium is present inside a cell? Yesterday, I was talking to you.
Endoplasmic
Endoplasmic
reticulum.
reticulum.
Yes, here it is also written that these calcium molecules, they are present inside this endoplasmic reticulum, and this endoplasmic reticulum that the calcium can come out of that endoplasmic reticulum through this calcium pumps, and this is your hydrogen pumps that is mostly seen in case of lysosome. You know, this lysosome is responsible for digesting all these things. All the enzymes are there, maybe digestive enzymes are there. So this is how the change of this ATPase, you see, to ATP to ADP and inorganic phosphate, how the hydrogen ion is coming out. Okay, so this is how these in lysosome it maintains low pH with a high concentration of this hydrogen ion concentration. Okay, so this is how this H plus ion is coming to this vacuum. Where is my pointer? Yes. And next, coming to this exocytosis. Okay, so in exocytosis, you will find that movement of large molecules bounded in vesicles are thrown out of the cell with the aid of ATP energy. Okay, see. So here there is a vesicle, and inside the vesicle the unwanted things are there, and it get attached to the plasma membrane and it is thrown out of the cell. Okay, so these waste materials, these neurotransmitters, hormones, mucus, polynucleotide, whole cells, polysaccharides, proteins, they can move out of the cell with this. Okay, now here you see the endocytosis. There are three processes associated with this endocytosis. One is called your phagocytosis, another one is pinocytosis, another one is your receptor mediated endocytosis. Okay, receptor mediated endocytosis. You see, phagocytosis is also known as cellular eating. Okay, and this pinocytosis is called otherwise as cellular drinking.
Cellular drinking.
Yes. So in cellular eating, this engulfs larger molecules like whole cells and bacteria. Okay. For example, the macrophages ingesting bacteria or own out red blood cells. Okay. And see how these unicellular organisms, they are also engulfing these food particles. Okay. Now this pinocytosis, this is also called as cellular drinking. Okay, so yes, you see how these molecules are taken, and see, this is called cellular drinking or pinocytosis. Okay, so you can expect one like objective question, 1 mark question from this. So these intestinal cells, the kidney cells, the plant. Yes,
Sir, pinocytosis occurs in amoeba.
yes. Basically, Basically, these organisms, those are living inside water bodies also. This pinocytosis occurs. Now coming to this receptor mediated endocytosis. No, see, so this is a process of endocytosis, but it doesn't occur on its own. Okay, it doesn't occur on its own. So for this endocytosis, you need some kind of receptors. Okay, some kinds of receptors are there, and that is utilized for this process of endocytosis. You see, these are your receptors. What are these receptors? Who can say? Yes, I was telling when we are dealing with this plasma membrane, I was telling regarding these extrinsic, intrinsic proteins and the role of proteins. So certain proteins, they work as receptors, right? So they can recognize certain ligands, certain molecules or certain compounds they can, or certain substances they can recognize. So these are called your receptors. They are present over the plasma membrane. Okay, so these are the plasma membrane. You see, the plasma membrane, the thickness of the plasma membrane is not constant throughout the cell. Okay, there are certain thin and thick sections, portions. The thin portions, we call them like pits. Okay, these are pits. So in these pits, sometimes these, so there are certain structures like pits, and these pits are associated with certain proteins. Okay, so they are coated with certain proteins. We call them like coat proteins. Okay, so the pits, those are associated with these proteins. We call them like coated pits, and these proteins are your clathrin molecules. Okay, so this is the clathrin molecule. Okay, so with the clathrin molecule, these pits are covered. So this is called a coated pit. Okay, so whenever there is certain substance that can be recognized by these receptors present over the plasma membrane, they can be taken inside the cell through these coated pits. See, now see these ligand molecules, they are moving inside these coated pits, and after certain times the invagination of the pit increases, and finally what happens, this pit or this coated pit, that is like pinched off from the this plasma membrane, and it forms the vesicle, and this vesicle, it is associated with certain other proteins. What are these proteins? We call these proteins as clathrin proteins. Okay, and these coated vesicles, then it moves to the lysosome through a particular path, that is then they form this early endosomes, then the late endosomes, and they get merged with this lysosome, where these things are digested. Okay, so that is happening through this receptor mediated endocytosis. We'll take one entire class regarding how the receptor mediated endocytosis occurs in a cell, and how these foreign substances are digested through this lysosome. What are like your early endosome, what are your late endosome? We'll discuss that in a separate class. Okay, today you just know that there are three kinds of endocytosis. One is your phagocytosis, pinocytosis, and another one is your receptor mediated endocytosis. Okay, and the example of this receptor mediated endocytosis is your uptake of cholesterol by the animal cells. Okay, so this is, see, this is a pit. Okay, so you see in the plasma membrane, I told you that all the section of the plasma membrane is not uniform. There are certain thin area. So this is called a pit, and in the pit, this pit is covered with certain protein molecule, that is called your clathrin molecules. Okay, so that pit is called a coated pit, and over this pit or over this plasma membrane, these receptor proteins are there. Okay, and these receptor proteins, they can recognize your ligands. Okay, and now this is pinched off from this plasma membrane and it forms a vesicle. Since this vesicle is attached with your clathrin protein, so we call this as a what? Coated vesicle. Okay, so this coated vesicle, it will form this, and inside the plasma membrane, these inside the your cytoplasm, these molecules, these clathrin molecules or this coat protein, they will be dissociated from this and it will form this early endosome where the pH is say around 5.5, something like that, or nearly 6, and then it will be converted to late endosome where the pH is still less, and it is again merged with this your lysosome. Okay, now you know what is phagocytosis? That is when the solid substances are taken or engulfed, and in case of these liquid things, that is taken with the water, that is called your cellular drinking. And whenever there is this use of this, so you see, this is your normal phagocytosis. These are your, so this is your phagosome. So this is solid substance that is taken inside, engulfed by the cell, and here it is taken, the liquid things are taken with the water. So that is your pinocytosis, and this is your receptor mediated endocytosis. Okay, now I was talking about this cell communication between cell and cell. So there are different processes by which the cell communicate with other cells or nearby cells. So there are different junctions, like in case of animal cell, you will find this tight junction, this desmosomes, and this gap junction. Whereas in case of plant system, you will find this plasmodesmata. Who can say what is a plasmodesmata? Anybody?
Connection between two cells.
In case of plants, right? Plasmodesmata is not there in case of your animals.
Two cells.
Okay. Why it is required?
Transport of one molecule to other.
So when there will be a transport between two cells, right? So when there will be a communication between two cells, you need a common path. So for that, this plasmodesmata is there. But in case of animal cell, you will find this tight desmosome, and there are tight junction, gap junctions also. Let us see. So this transmembrane proteins, opposite cells, attach in tight zipper-like fashion, and there is no leakage here. See, just like this, just like a zip. Yeah. So there is no leakage, and you can find this tight junction in the intestine, kidney, epithelium of skin. Okay. And these are your desmosomes. This is your desmosome. Okay, just keratin-like things, filaments are there made up of keratin. So this cytoplasmic plaques of two cells, they bind with each other. So this is your desmosome, and you can find this kind of junction in stomach, bladder, and in heart, because they always change their shape, right? When there is a lot of food in your stomach, it swells up, right? And it also sinks in absence of that. Okay, similarly the heart, when the heart is pumping, right, in diastole and systole, you have seen that the volume of the heart increases and decreases, right? So in that case, this stretching is possible due to the presence of these desmosomes. And coming to this gap junction, you will find this channel protein. We have seen this channel protein. So this channel protein, opposite cell joined together, providing channel for iron, sugar, amino acids, and other small molecules. See, this is one cell, this is another cell, and this is a like channel. Okay, so this is called your gap junction. You can find this gap junction in case of your heart muscle, this animal embryos. Okay, so this allows communication between these cell membranes. Okay, so, and in case of this plant cells, right, So the channel is between two cells. You know, in plant system you have your cell wall, right? Very rigid cell wall. So this plasma membrane of adjacent cells, they are connected with the channels, and this plasmodesmata that helps in connecting two cells. Okay, and through this, you know, this water is transported from root to the apex of the plant. Okay, how it is happening? So these cells are connected with this plasmodesmata, and it also helps in the transportation. Okay, so it allows the exchange of cytosol between adjacent cell, moving water from root to the apex, and the small solute, sugars, amino acids, and the example you can find in xylem and phloem in plants. So this is in total all about this plasma membrane and its function and certain processes associated with the plasma membrane, right? So you have a certain idea regarding this plasma membrane and these organelles. Of course, regarding these organelles, their function, those will not be coming in the exam. Rather, these plasma membrane things, those will be there in the exam. And from this portion, you can find only some short notes or short questions. Okay, and when I will be talking regarding this receptor mediated endocytosis, I will tell in detail regarding that, and that can come as a long question. Okay, so before that, we will be dealing with certain other things, and I will take some couple of minutes telling about that. I will ask you certain things. Can anybody tell where the protein is synthesized in a cell? Yesterday also I have asked this.
Ribosome.
Very good. And ribosome is present in the cytoplasm, right?
Yes, sir.
In the cytosol. I told you that the ribosome is present in two forms. Either it is in attached form with this endoplasmic reticulum, that is your raw endoplasmic reticulum, or it is present in free form in cytoplasm, right?
Smooth endoplasm.
Okay. Okay, very good. So the protein is synthesized in the cytoplasm, and you know that there are certain enzymes, those are present in mitochondria. There are certain enzymes, those are present in your chloroplast or these plastids, and there are certain enzymes, those are present inside the nucleus also, right? So all these organelles, these membrane bound organelles, are made up of proteins, right? And all the protein, those are present inside the nucleus or outside the nucleus or in the organelle, anywhere in the cell. All the proteins are synthesized where? In the cytoplasm. Okay, in the cytoplasm, with the help of your ribosome and your messenger RNA, the protein is synthesized in the cytoplasm. But how the protein is moving to different portions in the cell?
By the vesicle.
Okay, by the vesicle, that is not always vesicle is responsible, but there are certain other things by which the protein is transported to different locations. See, protein is synthesized at certain place, and then protein is transported to other place. See, protein is not a person or a organism or an organism that it will go to a certain place. So how it moves? Yes, there are several mechanisms by which it moves, but to a particular location, how a protein knows that, okay, I have to move to this Golgi body, I have to move to this chloroplast, or I have to move to this endoplasm.
Neural signal.
Okay, so when, see, I'm giving you a simple example. Suppose you have a letter, you are writing a letter to a person, right? You are sending that letter to some of your friends or relatives. How you send that?
So they have a signal sequence.
Very good. So how you send that? You just answer my question. How you send that?
Address.
Very
Address.
good. Very good. So what you need? You need an address. Okay. Without an address, can you send any message? Whether it is an email or a normal surface mail or anything, even a WhatsApp message, You need an address, right?
Yes.
Yes, sir.
What else you need? What else you need? Suppose you have an address. You have an address. Can you send that?
No,
Can
a
you send it?
carrier. You need
We need a messenger.
Okay. You need somebody who can transport that message.
Yeah.
Right?
Yes, sir.
So you need
Yes,
a carrier.
Messenger.
You need a messenger, right?
Yes.
Yes,
Yes,
sir.
sir.
Can you send the message? No. Why? Suppose you are sending a WhatsApp message, right? So if you have an address and you have a messenger like WhatsApp, can you send the message? No. You need internet. Yes or no?
Yes, sir.
Yes, sir.
Yes, sir.
You need internet, right?
Yes, sir.
In case of a surface mail, if you have an address and if you have one messenger, you can send this letter. Can you send this letter? No.
No, sir. We need internet.
You need certain system.
Yes,
You
sir.
need a postal department, right? And for that you have to invest certain amount of money, right? So in case of this system, in case of this organellar system, or in case of cell, how the message then can be or how the protein can be sorted to different locations. So in all the protein, they have a sequence that is called your signal sequence, right? You have a signal sequence in each and every protein, and that tells about the destination of that protein, to which location it should move, right? And again, you see, you have certain proteins, like you have seen these carrier proteins or channel proteins or different proteins are there. They can tell you, or they can give you the passage through which this protein can move, right? And the most important thing, just like your internet or your this postal department, certain amount of energy is also required, right? Certain amount of energy is required, right? So in case of a cell, what will give you the energy? The
Mitochondria.
energy molecules, those are there in the cell. Basically your ATP, GTP molecules, those are there, right? So the energy that will be utilized. So the protein through which so there are special kind of protein through which other proteins, they can move from one place to another. Those are present over the membrane. You see, in case of a membrane, suppose in case of mitochondria, it is a double layered membrane structure. Yes or no? So there are two membranes, and there is outer membrane, there is inner membrane. Suppose the protein need to move inside this matrix of this mitochondria. So the protein has to pass through the outer membrane, then to the inner membrane, and then to the matrix. Right? So there must be some kind of thing that is present over the membrane that can allow the passage of these proteins. So those passage, we call those proteins, we call them like translocon. What we call? We call them like translocon. Okay. So the process, the entire process by which the protein is localized to a certain destination in a cell, okay, we call this translocalization of protein or translocation of this protein. How the protein is transported to different locations in the cell. Okay, thank you for joining today. We will discuss this protein localization in the next class. Okay.
Thank you, sir.
Thank you, sir.
Welcome.
Thank you, sir.
Thank you, sir.
Thank you, sir.
You can quit the meeting right now.